Welcome back to our video on neuroinflammatory pathways and disease. We'll be looking at the interaction between the brain and the immune system and how this contributes to the onset of neurodegenerative and inflammatory conditions. First, we've got to start from the blood-brain barrier, separating the brain from the immune system. It is a semi-permeable membrane that separates the circulation from the interstitium of the brain. It is a triple-layered structure formed from endothelial cells bound by tight junctions containing pericytes and astrocytes as end-feet processes and the capillary basement membrane. Here you also have numerous scaffold proteins and transmembrane proteins such as oculodin, claudin, tricillin, and junction adhesion molecules that really reinforce and solidify the blood-brain barrier, stopping immune passage into the CSF. The blood-brain barrier acts to filter and regulate entry of substances and protect from harmful substances, providing a selective diffusion and paracellular barrier. Looking at the functional role of the blood-brain barrier, it's crucial to identify that it's selectively permeable, allowing nutrient transport of essential nutrients such as glucose via transporters and amino acids, while also removing metabolic waste products from the central nervous system. It acts to protect against pathogens and entry of most pathogens, and it's important for the regulation of ions, maintaining ionic composition, crucial for neuronal functioning and generation and propagation of action potentials. It allows for immune surveillance through controlled passage of immune cells, for example T cells, for immune surveillance without triggering an inflammatory response. Immune interaction with the blood-brain barrier takes form immune cell trafficking. First, you have surveillance. Through the patrolling of immune cells, such as T and B cells, monocytes, which monitor the CNS for signs of infection or injury. Immune cells utilize specific adhesion molecules, for example, selectins and integrins and chemokines, to adhere and transmigrate across the blood-brain barrier, which is important for creating a chemotactic gradient to direct and recruit more lymphoid tissue. It allows for selective entry into the blood-brain barrier, with only activated for specific subsets of immune cells that can cross the blood-brain barrier, typically in response to infection. The blood-brain barrier creating this immune privilege for the CSF limits the immune response, where the immune responses are tightly regulated to prevent excessive inflammation that could damage the neural tissue. Immune regulatory cells such as T regulatory cells are important in suppressing excessive immune responses and through the release of anti-inflammatory cytokines such as interleukin-10 and TGF-beta, which help maintain the immune homeostasis. Neuroinflammation and its processes. Separate to systemic inflammation, intrinsic inflammatory responses in the central nervous system respond to injury, infection, and disease to maintain neural function and protect against pathogen. Activation of innate immunity mechanisms in glia, or activation of microglia, causes release of cytokines, presentation of antigens, and phagocytosis of harmful agents, and astrogliosis, or formation of reactive astrocytes, neurons, and microvasculature. Cytokines and chemokines are important inflammatory mediators such as interleukin factor 1 beta, tumor necrosis factor alpha, high mobility group BOX1, transforming growth factor beta, and prostaglandins, which can alter neuronal, glial, and blood-brain barrier functions, for example its permeability to immune infiltration, by activating transcription and post-translational mechanisms in neurons to propagate the immune response and recruitment of adaptive immune cells. Aging has been shown in research that a healthy but aging brain has chronically increased levels of pro-inflammatory cytokines and reduced levels of anti-inflammatory cytokines, demonstrating that the factor of age alone is linked with chronic neuroinflammation. Further research has uncovered that aging brains also have an increased number of activated microglia, a sign of activated immune system response, demonstrating another link between aging brain and neuroinflammation. It is believed that those with a disease such as neurodegenerative conditions have an abundance of activated microglia that can phagocytose amyloid beta, which can lead to the continued plaque accumulation. Neuroinflammation is seen as a major component of Parkinson's disease, where it is believed that inflammatory responses in the gut may be linked with the initiation of the disease, leading to inflammation in the brain, particularly of the substantia nigra, and disrupting the production of dopamine, which is characteristic of the disease. Lastly, neuroinflammation is considered to play a major role in initiation and progression of multiple sclerosis, where research has shown that the blood-brain barrier becomes disrupted by the presence of inflammatory cytokines, which allows for B cells and plasma cells to enter the central nervous system. Research has shown that the blood-brain barrier becomes disrupted by the presence of inflammatory cytokines, which allows for B cells and plasma cells to enter the central nervous system, where they damage the myelin sheath covering neurons, and this demyelination is a major symptom of the disease. An emerging theory is that stress plays a key role in initiating the deregulation of the immune system in psychological conditions. Alongside genetic, epigenetic, and environmental factors, the immune system activation has been seen to cause abnormal neurotransmission, resulting in serotonin deficiency and the increased production of neurotoxic substances that heighten disease progression. Illnesses such as schizophrenia, autism, and depression, and other mood disorders have been linked with inflammation of the brain. However, the exact underlying mechanisms of the relationship are specific to each illness. Let's break down the overlying process of autoimmune disorders and how the immune system is mediated to generating all these inflammatory responses. Antigen presentation, where autoreactive T cells recognize CNS antigens presented by professional antigen-presenting cells, more commonly dendritic cells. These T cells then become activated and acquire the ability to cross the blood-brain barrier. Cytokine release from activated T cells, such as interferon gamma or tumor necrosis factor alpha, weaken tight junctions, and T cells and other immune cells produce MMPs that degrade the basement membranes. Upregulation of adhesion molecules, for example ICAM1 or VCAM1, on endothelial cells facilitate immune cell binding, where immune cells traverse the blood-brain barrier via paracellular or transcellular routes, and the activation of resident cells by these infiltrating immune cells activates microglia and astrocytes. Resident cells release additional cytokines and chemokines, attracting more immune cells and amplifying the immune response. So how does this process lead to neuronal damage? Firstly, you have direct cytotoxicity, where immune cells and inflammatory mediators directly damage neurons and glial cells. You can also have demyelination, with loss of the myelin sheath impairing neuronal signaling. Cytokine signaling, for example, through interferon gamma, tumor necrosis factor alpha, promote expression of adhesion molecules and increase the blood-brain barrier permeability. Interleukin-17 by T helper 17 cells contributes to inflammation, and again, this disruption of the blood-brain barrier. Adhesion molecules, selectins, and chemokines play a similar role in this immune response for immune cell recruitment, mediation, and integration into the central nervous system, which then amplifies up the response from these chemokines to damage neuronal tissue and result in the subsequent demyelination commonly seen in conditions such as multiple sclerosis.